Our proposed treatment for articular cartilage defects involves using an organ-on-a-chip platform to create an ideal environment for growing transplantable articular cartilage, which can then be surgically implanted into patients with osteoarthritis, specifically osteoarthritis of the hand and wrist, for which there are fewer existing treatments that restore close to full function. My name is Maya Jenks, and you will also hear my co-presenters, Victor Ma and Tori Nelson.
The first part, I would like to discuss about the technologies of our products. So our approach runs on three very simple steps shown below. So the first one, step one, collect. We draw a teaspoon of the bone's marrow with a local numbing, chill the tube on ice, and carry it straight to the lab. And next is step two, isolate. A quick centrifusion spin and short plastic adhesive steps, both pain-free, gently pull out the fresh meso stem cells while keeping costs low. Step 3, seed. Those cells enter microfluid chips that provide soft fluids and growth cues. They organize, lay down tissue, and pass on chip quality checks. The various stem cells moved into the phase 1 safety trial. Small harvest, simple probe, smart chips, bring the autologous stem cell therapy close to the patients. Clinic impacts shows the values of our products. Here we list three main clinic impacts we came out. The first one is adding the regenerative tricks, like the stem cells could put off the needings of big surgeries, save the resources in hospitals, and letting the joints heal longer, and widening the lives, which will reduce the suffers from patients. The next one is hospitals that jump on these technologies can set up the lively orthopedic innovation hubs, attracting express fundings and faster patients breakthroughs. The last one is combining the MSCRs with the PRP SIMs to cut the pain and boost movements based on the knee-associated studies extending to other joints, including the product we are doing right now.
Total market for products like ours is the 1 in 5 U.S. adults with osteoarthritis, a subset of whom have radiographic osteoarthritis of the hand. Within that group, our target market is patients whose hand OA is symptomatic and not sufficiently treated by supportive interventions. For these patients, the new treatment will address the limited supply of transplantable cartilage and provide better functional outcomes than joint fusion or replacement. A potential barrier to entry is the necessity of new infrastructure in order for hospitals to implement this treatment, such as facilities for culturing the OOC-grown tissue. However, the osteoarthritis market is growing, and we hope to be able to offer this procedure at a similar price to the current standard of care. In bringing our product to market, we would like to license existing patents, including aspects of existing organ-on-a-chip platforms used in drug development, as well as particular hydrogel formulations to support cell growth. Implementation of the treatment would incorporate certain surgical tools and techniques under existing IP agreements with the treatment facility. We then would look to patent the novel aspects of the technology, including the specialized OOC platform, implantation techniques, and combinatorial approaches for promoting tissue growth.
For regulatory, because our technology presents a significant risk to the patient, the FDA would consider it a Class 3 device. Our technology is not only essential to the treatment of articular cartilage defects, but also poses an infection risk, ultimately affecting the health, safety, and welfare of the recipient. Since it is a Class 3 device, we cannot file for a 510(k) exemption, so we'll have to go through the pre-market approval process. As such, we will need to apply and maintain an investigational device exemption, IDE, in order to conduct research demonstrating the efficacy and safety of our technology. These are the two main criteria the FDA uses to determine market approval. We're planning on combining two business models, capital equipment and pay per use, in order to distribute and sell our technology. First, we'll adapt our OOC platform into a piece of capital equipment that healthcare facilities can purchase. We are also planning to train and provide an on-site technician in order to operate the equipment and charge a fee for each use. As such, we're aiming to use a no-frills service model in order to reduce costs associated with clinical support while also seeking to capture new market share. In terms of reimbursement, we're hoping to replicate the process for autologous chondrocyte implantation ACI, a procedure where chondrocyte cells are extracted, expanded in vitro, and reimplanted back at the cartilage defect site. Both the procedure and materials are reimbursed in this instance. For ACI, the procedure is reimbursed using CPT code 27412 with a payout for Medicare of approximately $1,700. This can vary across regions and annually. The materials used to extract, grow, and implant the chondrocyte tissue are reimbursed through HCPCS code J7330 with a maximum payout of $21,000 from Medicare. Ideally, we would price capital equipment and set fees with reimbursement for an ACI procedure in mind, while also taking into account our early development expenditures. We wanted to take a minute to introduce ourselves and thank you for your attention during this presentation. We very much enjoyed collaborating together on this project and look forward to answering any questions. Thank you.